Hello, and welcome to our discussion of neurons. We are going to be going over the anatomy and electrical communication. Here are the associated readings, both in the Brain Facts and OpenStax texts, and you'll notice that it's taking pieces of both that will both clarify and be supplemental. How important is what we're doing? Well, when we look at the general knowledge of neuroscience, we find there are a lot of myths that people believe. So, a big one, the most believed myth at this time, is about learning styles. Learning styles is the idea that, oh, I'm a visual learner, so I can only learn from slides or videos or whatever it is, or I'm some other kind of learner, so I need it to be presented in this way. Utter nonsense, and yet widely believed by the public, teachers, and neuroscience students. All this learning styles nonsense does is it makes it less likely for you to be successful because your biases go into a situation. You're like, oh, my learning style doesn't match. I can't learn. The Mozart effect. Does Mozart make us smarter? No, absolutely not. There was one study done at UCI years ago that showed that if you listen to Mozart, you got a small boost for like 15 minutes, a very brief time, performing certain cognitive tasks. And all of a sudden, there's many videos for babies to make your baby a genius. Pseudoscience, nonsense. Children are less attentive after consuming sugar? Also untrue, and yet most people believe it. Left brain, right brain is something we'll explore in greater depth. The idea that you are either left-brained or right-brained, which again, no basis in science, not testable, and meaningless. Basically, it's using the brain to discuss personality types without any evidence. And the whole 10% of your brain notion. 14% of neuroscience students believe this? That is just upsetting. You use your whole brain. Your whole brain is active. Every neuron in your brain in the last few seconds has done something. So saying that you only use 10%, it's a meaningless number. And no one knows where it came from. But people do quote Einstein on that, which good luck finding the quote. Well, let's get into what neurons are and how they work. The first thing we consider is we've got these two kinds of cell in the brain. We've got neurons and glia. Glia, there's as many glia as neurons, and they do a lot of important tasks. We've spent so much time historically focusing on neurons because neurons were easy to study, comparably, and they weren't even human neurons. A lot of what we know about neurons comes from the study of squid neurons. Squid have giant neurons, so big you can see them with the naked eye and work on them directly with a microscope and basically working on a live neuron that is removed from a squid in action. So that's where a lot of this stuff comes from. Glia, much more difficult to study. The human brain contains about 86 billion. Different books say different things. One book says 86 billion. Okay, cool. It's somewhere in that range. Interestingly, the majority of those neurons are in your cerebellum. Your cerebellum being a fairly small part of the brain, which means you have a part of your brain that is just densely packed with neurons compared to the neocortex. So let's take a look. Now these neurons here are about your central nervous system. You also have neurons in your hands, in your fingers, in your toes, everywhere in your body, because anywhere you're sending information that gets back to the spinal cord is going to be along neurons. However, peripheral nervous system, hand neurons, are going to look and function differently than brain neurons. Not completely differently, but enough that we want to consider. Now here is the cerebellum in purple in this lower right portion of this brain. Comparably small, yet containing the vast majority of your brain's neurons. We also see the cortex, right? The corpus callosum connecting the two hemispheres. Basically, each hemisphere is functionally connected to the other one through this corpus callosum. Otherwise, quite separate in the individual tasks. Yet, for all of us with a functioning corpus callosum, it's all tied together. And this image is nice because it gives us, it's a little interesting. The middle of the brain it's kind of showing a lot of different parts. The middle of the brain is where you see corpus callosum and thalamus. So right there, we're near the center. Around there, we have the hippocampus, which is actually kind of in the temporal lobe. So not really in the middle, not on the side, embedded in the temporal lobe. And then the wrinkly parts are the cortex, and those are the surface of the brain. So this image is kind of showing us where things are in general. And navigating through the brain is going to be something we're going to spend some time doing. The two major delineations in the nervous system are the central nervous system and the peripheral nervous system, with the central nervous system being your brain and spinal cord. Your peripheral nervous system being your everything else, right? Cranial nerves and spinal nerves. And we already mentioned that things are going to function differently within there. We know what we know because starting with Santiago Ramon y Cajal in the late 1800s, he built his own microscopes, had incredible vision and an ability to see neurons, and using a recently discovered staining method was able to look at individual neurons and see their components and connections. Cajal's drawings, which we see an example of here, are so good that there are modern equivalents, but there's nothing better. In terms of like a hand-drawn artistic rendition of a neuron, the guy was a fantastic artist. He used the microscopes that he built, and he showed, the first person to show, that the neurons were separate and did not grow into each other. And his Nobel Prize speech was arguing with the guy who invented the stain. They shared the prize about whether or not the neurons grew into each other. And Cajal gave us this neuron doctrine, which was these neurons are functionally separate pieces. It wasn't one big thing, right? That we're all kind of connected. They are connected, but these spaces between neurons are interesting. And we'll talk about that. That next segment is all about the connection between neurons. This tiny, tiny space, almost invisible. The ability of Cajal to see this, again, baffling, right? Because if you were to look at neurons under a hand-built microscope and try to draw them, incredibly difficult. If you take the lab and do some looking at the histology yourself, it's not like this beautiful picture here, right? Very clear, artistic rendition. Neurons generally aren't this pretty. This is also a peripheral nervous system neuron, and we need to understand the difference. How we know it is these cells wrapped around the axon, right? The axon being this whole thing covered with these yellowy cells over the purple, right? From the axon hillock here, near at the edge of the cell body, all the way to the axon terminals. This whole thing is the axon. And these things covering it are called Schwann cells, and they only exist in the peripheral nervous system. They create myelin, and myelin is an important part of the neuron and how it functions. Now let's look at the differences in terms of these neuroglia. Basically, the glial cells produced by these different parts of the nervous system do different things. So on the left, we see the central nervous system glial cells, and on the right, we see the peripheral nervous system. The ependymal cells are interesting, but we're mostly interested in the difference between the oligodendrocytes and Schwann cells, because the oligodendrocytes are part of the central nervous system, and they create myelin in the central nervous system. The Schwann cells are part of the peripheral nervous system and create myelin there. Satellite cells and astrocytes do approximately the same thing, with satellite cells giving support in the peripheral nervous system, and astrocytes having this incredible duties in the central nervous system. Microglia being the immune cells in the central, where in the peripheral you have your typical immune function. But the central nervous system is very specific. So we look at the central nervous system glial cells. And what we see is that you've got these astrocytes that do a lot of tasks. They hold the neurons in place, they envelop the synapses themselves, and they actually can release neurotransmitters, absorb neurotransmitters, and in some ways, they act as conductors for groups of neurons to keep a bunch of neurons firing in sync. And a lot of what we know perceptually and believe is that this synchrony is what allows neurons to function the way they do. Right? It's what allows us to function the way we do. The myelin is a fatty substance that insulates the neurons, and these tiny little spaces in the myelin for myelinated cells allow these neurons to basically fire faster, to send a signal along the neuron faster than if they did not have myelin. When you look at a brain, the brain it's fresh. The brain is white and pink. The pink is mostly blood and sometimes a little more grayish, kind of like the gray matter. But it doesn't look like, the kind of peachy colored brain that we look at, like we have like a brain model. The brain is basically this bright white because of so much fat. And then your ependymal cells mentioned, they line the ventricles and they produce the cerebral spinal fluid. So they're interesting, but they're necessary for the structure, but less so in the function. Now, of the nervous system. Neurons have the same anatomy as any other cell, except for those two big pieces that really make it special, the axon and the dendrite. So all we're looking at here is your traditional cell. Previously, we discussed how proteins and DNA, DNA produces proteins and how that's important. And so what you're looking at, your neuron has all these things, and these things include the ribosomes. The ribosomes are going to be producing proteins as your neurons constantly adapt and change. You also have in the cell wall specific types of protein channels. Now, your cells have protein channels, right? Things get in and get out. But in neurons, you've got a couple types of very special proteins embedded in the cell wall that allow for ions to move back and forth so that a signal can go from the income from the dendrites to the cell body through the axon to do something at the synapse. The motor neurons, they have their soma in the spinal cord, and they receive excitation from other neurons and conduct the impulse along its axons to a muscle. So, here, my hand. Right? These are muscles in my hand. If I want to bend my fingers, I have to send a signal from my spinal cord that says, bend your fingers. So sensory neuron is specialized at one end and gets input. So basically, if I touch something, I am doing something to a neuron, a sensor in my fingers that are connected to a neuron that are sending it, or part of that neuron that are sending it to my spinal cord. And if something happens, and it comes back here and to do something. We'll talk about the reflexes and things. We talk about the synapse and how these things work. The idea is all this information is going back and forth for processing. And your spinal cord, having 1 billion neurons, is doing kind of what the brain does, but stuff that, again, we don't have to think about. Now, dendrites are these branching fibers that receive the incoming info. So you've got a synapse forming between the dendrite of one neuron and the axon terminals of another, and then a tiny, tiny space between them where neurotransmitters are going to cross. Dendritic spines are growths on the dendrites that increase surface area. The more dendrites you have, the more surface area you have, the more synapses you can form. And each neuron is forming thousands, or is at least capable, especially in your brain, of forming thousands of synapses with other neurons. A lot of information. The soma, cell body, same as everything else. It does the metabolic work, but the cell wall of the soma also, cell wall, the cell wall of the neuron is allowing charge to pass through. So it's still doing the same kind of tasks as the rest of it. And then, of course, our axon, where you have the fiber transmitting the signal, and your axon terminals are connecting to possibly thousands of dendrites. And in our peripheral nervous system, these nodes of Ranvier, right, or in our central nervous system where we have myelinated axons, they're just using oligodendrocytes instead of Schwann cells, allow the signal to jump. So here's a little preview of what we're going to be looking at. We see here the difference between the central and peripheral nervous system. Here your oligodendrocytes can do more than one section. Here in the peripheral nervous system, they only do one. The space between them allows the signal to jump. So instead of having to open channels all the way down, you're going to get the signal that opens channels here, and then here, and then here, and then here, instead of opening all these channels. Saves energy, saves time. And that's the key, right? That's the point of myelination. The presynaptic terminals are the space between, at the very endpoint of the axon. So there's two terms here, presynaptic and postsynaptic. And so we look at this red arrow, and we're looking at, okay, here is the space between them. It's almost invisible. It's basically pixels wide, if we're looking at the image here, between the axon of here and the dendrite here. They're not connected, right? They're not touching, but the space is tiny. So you're looking at the signal coming here, opening up, releasing neurotransmitters, and jumping to here. And again, we're looking at how these signals are sent along the neuron. Well, a couple of terms, afferent and efferent. These are nervous system terms that have to do with the direction of information flow. So you consider neurons can be both, because they've got an afferent part, which receives information, and they've got an efferent part that sends it out, dendrite, axon. But you've also got neurons that would be considered afferent neurons, sensory neurons, bringing information in. Motor neurons, efferent, bringing information out, sending information out. And what we think about here is A and E. A comes before E, and the way I remember this is you have to sense it before you can react. You have to sense it before you can react to it. So afferent, sensory, efferent, motor. So spinal cord, if you think about a reflex, you touch something hot, afferent, this is hot, pull your hand away. Hot signal. Interneuron in your spinal cord, processing it, sending some information up to your brain, and some information immediately to a motor neuron saying, pull your hand away right now so you don't burn yourself. So we have all these things working like this. I showed you pictures of these kind of artistic neurons. I showed you something that Cahal drew. Now we're looking at some very different types of neurons. The idea being they are structured to relate to their function. So depending on what a neuron does, its structure will be different. And that's the key. They all got axons, they all got dendrites, but we are going to be looking at a number of different functions. In fact, the Cahal drawing we saw is this one in the middle, similar to this one. This is a Purkinje cell. Cahal's drawing was, I believe, of a pigeon's cerebellum. They look just like ours. These are responsible for myelination, the white matter in the brain. And so this is the kind of question we think about. Do you remember what it was? Well, it can't be neurons. So we're kind of left with astrocytes, oligodendrocytes, and microglia. Well, it's going to be the yellow, right? These oligodendrocytes are the ones that have the glia producing in the brain. And if you recall, in the peripheral nervous system, you're looking at your Schwann cells. Most of a neuron's membrane is phospholipid molecules, but it also contains what? What else is in the membrane? Well, the answer is it protein molecules, neurotransmitters, dendrites, or axons? Protein molecules. And that's the key, right? That allows ions to move in and out. The nerve impulse travels away from the cell body along the what? Soma, dendritic spine, dendrite, or axon. And the answer is away from the cell body, right? So information's coming in, cell body, and going out. So going out, away, is the axon, right? The efferent part, if you will. Now, the blood-brain barrier. When you hear a term like this, it sounds like you've got like a wall built between the blood and the brain. But that's not entirely true, because your brain is filled with capillaries. There's blood everywhere. The issue is, in most of our body, things can flow in and out through our capillaries directly into our tissues. It allows the fast movement of nutrients. However, the key is our brain, while it shows incredible plasticity, an incredible ability to change and adapt, once we hit a certain age, we are probably not producing new neurons. While there's debate about this, if we are, it's very few. So if you have something that's attacking your skin or an organ, you think of maybe a sore throat or bronchitis, you have a bacteria or a virus attacking your body. It causes damage, and then your body repairs itself because we can produce new cells. Your brain cannot. So your brain cannot produce new cells, which means you need an extra layer of defense to keep these things out, which most pathogens cannot cross in. So you've got these thicker endothelial cells in the capillaries, and you've got extra layers of protection, but it costs. So the blood-brain barrier, we're looking at it something like this. We see these cells around here. This in the middle is the vessel, the blood vessel. The lumen is kind of like this open space in there. There's no solid tissue, and that's where blood is flowing, right? And your blood vessel wraps around the blood, and you've got blood cells flowing through here. Now, in most of these, you see where these, the green is the astrocyte pushing it together, which allows very little flow, okay? Which means only fat-soluble chemicals can pass through, and water-soluble chemicals cannot. Water-soluble chemicals include things like vitamins, right? Some of them. Glucose. Right? Glucose, our brain runs on sugar. Certain hormones, which amino acids, which means you have to move things into the brain. This creates a cost. This also creates a problem for, modern medicine. Chemotherapy for brain cancer often has to be, injected through the skull or some other way because it can't cross the blood-brain barrier. So only things that can, so drugs, what can cross it? Drugs, that people take for both psychiatric things, right? Those can cross the blood-brain barrier, and drugs that people take to, get high, can also cross the blood-brain barrier. But for these other things, there's a cost, which means you have to use energy to get energy. Now, fortunately, one glucose molecule gets you a lot of energy, but there is still this importance of protecting the brain, and that's the point of the blood-brain barrier. Our neurons depend on glucose. This is called active transport. We need thiamine, which is a vitamin B, to do it. And without this, you can see the death of neurons. Poor nutrition, right? Either through basically alcohol use that can block the function of this vitamin, or through something like severe dietary restriction can cause this, which is really quite a massive brain damage, specifically in the cerebellum. Primarily in the cerebellum and hippocampus, which means cerebellum, your movement is now impaired permanently, and the hippocampus, your memory is now impaired permanently. And 20% of your brain's oxygen and energy is consumed, or your body's oxygen and energy is consumed by the brain. So your brain is this special organ that gets a lot of protection and attention. How do we communicate between neurons? Calcium channels, neurotransmitters, potassium channels, or blood flow increases? And the answer is neurotransmitters. Now, calcium and potassium channels are all interesting and important for the neuron to send a signal, but it is the neurotransmitters that cross the synapse. What percentage of oxygen is used by the brain? 10, 20, 30, or 50? And if we recall, that was 20. So how are we? Let's get into the impulse now. This is the key. We get the general anatomy of the neuron, right? The axon and the dendrite and the axon. Now we're looking at how is this signal sent. And so you look at this picture now, and if you've never seen a picture like this, you're probably going like, wow, there's a lot going on here. So we're going to be unpacking this. These little things in here represent ions. And so we see our 3 different types that we're focused on, right? We've got K+, which is potassium, Cl-, which is chloride, and Na+, which is sodium. Chloride is fine. We've got plenty of chloride, but it's not the interesting ones. Sodium and potassium are the 2 that we're going to spend time with. Now, the nerve impulse is the electrical message traveling down the axon of a neuron. It's going to be doing this by using sodium and potassium. When you're myelinated, it doesn't travel directly. It regenerates. And the speed of the nerve impulse ranges depending on myelination, type of neuron, less than 1 meter per second to 100 meters per second. And this translates roughly to 2 to 220 miles per hour. So some neurons can send a signal extremely quickly, and others are far slower. Let's first look at what in the world an ion is. Well, there's 2 main types, cations and anions, right? And you've got these atoms. What we're looking at here is hydrogen. 1 proton is a hydrogen atom. And so if you have a proton, which has a charge of 1, and an electron, which has a charge of -1, you have a neutral atom. There's no charge to it. If you have a proton and no electron, now you have a positive charge. You have an ion. If you have a proton and 2 electrons, you have a negative charge. You have an ion. So what makes this happen? Well, the idea is you have certain ions or atoms that want different things. Why does, for instance, salt, NaCl, sodium chloride, why does sodium chloride happen so easily? Well, the answer is sodium by itself, a pile of sodium, wildly explosive. It's got these extra electrons. It has to do with the electron kind of ring. So you have these outer rings that can be full or not. And you want to kind of reach this stage of, stability. So we look at the ions we have, right, these things. So sodium has an extra electron, right, basically 1 extra electron in its outer ring. This creates instability, and it wants to get rid of it. A pile of sodium could explode very easily. Chlorine. Chlorine is a toxic gas. It wants to pull electrons from things because chlorine is also unstable, but chlorine has an almost full outer shell. It's missing 1, right, in order to be full in nature. So sodium and chlorine, they love each other. They get together like so easily because sodium's like, hey, I want to get rid of this electron and have a positive charge because now I'm stable. Chlorine wants to grab an electron. It has a negative charge, which means you've got positively and negative charged, basically atoms, ions, in fluid. You put salt in water, it's not dangerous to you because your sodium has gotten rid of that electron that the chlorine has picked up, making the sodium positive and the chlorine negative. And hopefully this is much chemistry that I ever have to do. Now, imagine a system that says, let's take advantage of these differences in charges. What if you could have a wall here, and on this side of the wall, you pump out all of the sodium, and on this side, you pump out all the chlorine over here. So now you've got positively charged sodium ions and negatively charged chlorine ions. Well, you got a couple interesting things that are going to happen. One, you've got a charge difference, which means you're going to have an electrical gradient. And two, if you were to open a little space between those in that wall, your sodium wants to diffuse and your calcium wants to diffuse, right? They want to reach equilibrium. So you've got these two gradients working. You've got electrical gradient wanting to be neutral, and you've got concentration gradient wanting to also be neutral. Now here's the thing. Chlorine, we don't care. Chlorine is not going to be doing anything. Sodium and potassium are the two that we care about. You see, we've got the same kind of charge. We've got basically more negative inside than outside. But we're not moving the chlorine. The chlorine's just there. What we're doing is moving the sodium and potassium, and they're both positive. And this is the part that I think just baffles people. Because what we're looking at are two positive ions, and yet we're creating this charge change between the inside and the outside. So we're going to have to look at how this happens. So, as a neuron, this is going to be the overview. This thing in the middle is the charge as a neuron fires. It goes from rest, which is negative, to very positive, and then to extra negative, and back to rest at negative. You have these purple things, right, this, that represent potassium, these green ones that represent sodium, and these potassium channels and sodium channels. And what we're looking at is this. Sodium channels open first. We look at rest. More sodium outside, more potassium inside. So, it's more negative inside than outside. What does sodium want to do? Well, if it's negative inside compared to outside, which means these sodium ions are both positive and have less concentration inside, they have two forces pushing them inside. But if the channels are closed, they can't do anything. You open a channel, sodium rushes in. All of a sudden, you overshoot, you get this extreme positive charge. You open your potassium channels, your potassium rushes out. Now you've got this imbalance, and you've got another process that's going to slowly get rid of the potassium, the sodium, and bring in the potassium. And this has, again, a energy cost. These don't, because sodium wants to come in at rest. It's much more concentrated on the outside. Potassium's just going to chill. Potassium has two forces that are creating a kind of equilibrium. And the reason is, it's more negative inside than potassium, and potassium's positive. So it's like, well, this is good. However, there's more potassium inside than outside, so it wants to get out. Two gradients. Think of a drop of food coloring in a glass of water. If you drop it in and leave it long enough, at the beginning, you get this high concentration of color, and that's going to diffuse. It wants to reach this equilibrium. Wherever you have high concentration, it wants to flow to low. If you put a membrane around it, it can't move, but it wants to. As soon as the membrane's gone, it's going to diffuse. That's how this works. Now, in the peripheral nervous system, a single incoming signal can cause a neuron to fire. These are things like reflexes, muscle movement. In the central nervous system, there may be thousands of conflicting messages. We consider firing rate changes more than simple on and off. This is the part that, again, it's kind of confusing because you think about it. I want to bend my finger. Boom, signal. I want to imagine a sunny day at the beach. How many neurons are firing to make that image happen? And not only are you having neurons that say, stimulate this, you've probably got neurons that are saying, don't think about clouds. Don't think about rain. Inhibit these ideas. So you've got these messages that are conflicting. So many neurons working to create this experience. Now here's another image. And now remember, if you look at this image, you should be able to now look at it and say, are we in the central nervous system or the peripheral nervous system? Because Schwann cells are peripheral nervous system cells. So here we see how myelin works. You got these little nodes of Ranvier where these channels open. So if you don't have myelin, you have these ions diffusing everywhere. You open, you reach this threshold and the neuron sends a signal. It opens these what we call voltage-gated channels. Enough sodium comes in. You open enough sodium channels here in the dendrites in the body, and all of a sudden you reach this point here where it's so positive that you open your channels, your voltage-gated channels. And sodium rushes in. Sodium diffuses all the way to here. Now there's a limit to how big this space can be. You can't just diffuse all the way to the end. It would take too long and it might just not even work. But it diffuses to here, causing voltage-gated ion channels to open, and more sodium rushes in. Sodium enters, diffuses, rushes, here, opens more channels. Channel opens, diffusion. Channel opens, diffusion. Channel opens, diffusion. That's how the neuron is firing. Here's a bigger image of what we saw. We see these numbers here are different for every neuron, but the idea is you're more negative inside the neuron than outside. What this is an electrode that is measuring the membrane potential of the neuron. Basically, what's the charge inside? At rest, it's low, and you can have little stimuli that boost it up, but you need to reach the threshold in order for the neuron to fire. Once you reach this point, you open those voltage-gated sodium channels rapidly, which causes this huge rush of sodium into the neuron. Once you reach the peak, you open your voltage-gated potassium channels. So now potassium wants to get out. You've got too much potassium inside, and it wants to get out. Your sodium has now kind of overshot, and you reach this refractory period where you've lost so much potassium that you're even more negative where you were, and then you start to rebalance, right? Polarization is away from your resting state. So polarization means you're very negative or very positive, but we call this depolarization as we reach positive, and then repolarization as we go back to our resting state. So we're trying to build, and what I want you to do is to build an image in your mind of what this looks like. It's incredibly complicated because of all these moving parts. You have these at rest. We're looking at rest. The sodium channels are closed and potassium channels are partially closed. Now, why are potassium channels partially closed? And if you recall, it's this. Potassium wants to get out because there's a high concentration of potassium inside. Potassium wants to get in because it's more negative and potassium's positive and wants to diffuse where it's negative. So potassium at rest has two forces working on it, which creates this equilibrium. It wants to get out, it wants to stay in, it's confused. Sodium just wants to get in because there's less sodium inside and there's a negative charge inside. Positive wants to flow to negative, high concentration wants to flow to low concentration. Sodium channels are therefore closed. Potassium channels partially closed. Now, we also have this sodium-potassium pump that's constantly working to get sodium out and potassium in. This creates the gradient. So when you're at rest, you're pumping the sodium ions out to create more positive outside, and you're pulling potassium in. They're both positive, but the way you create this gradient is by for every 3 positive sodiums you pump out, you only pump in 2 positive potassiums. And so what this does for us is it means we are constantly getting rid of a little positive charge, maintaining the inside negative and the outside positive. Here's an example of what we call diffusion, right? Or something like osmosis when we're specifically talking about water. So here we have an example of different concentrations. Well, the liquid wants to go from to create the same concentration before. So osmosis can actually cause this odd difference in the height of these two things across this membrane because it wants to create the same concentration. In some ways, we can think of how does water it'll soak onto you touch a dry towel to water, it'll start to come up, right? Now, at rest, potassium is a higher concentration inside, wants to go out. Sodium wants to get in, but can't. All right. At rest, the charge inside the neuron is usually more what than outside the neuron? Positive or negative? The answer is negative. This allows us to take advantage of those gradients. The sodium-potassium pump removes how many sodium ions for every how many potassium ions? And the answer is, do we remove 2 or 3 of each? Bring in 2 or 3 of each. And the answer is yellow. We remove 3 sodium and bring in 2 potassium. When the neuron membrane's at rest, sodium channels are what? And potassium channels are what? The answer is sodium channels are closed and potassium channels are partially closed. Here is our action potential. We have this depolarization and repolarization. And here is our sodium-potassium pump constantly working. Now, can you look at this image here and say, what is happening? Where are the sodium channels opening? Where are the potassium channels opening? These voltage-gated channels are cool. Basically, positive enough, boom, fire. This is part of the all or none law, which is a neuron either fires or it doesn't. You get a little stimulation, not enough to reach threshold, nothing happens. So we've got our hyperpolarization, which is maybe you get a signal from another neuron that says, don't fire, extra don't fire, whatever you do, don't fire. It causes you to go. Depolarization, you have this opening up these sodium channels. And then when you reach your action potential peak, you open your potassium channels to get you back to a negative rest. Your threshold being whatever minimum you need in order to cause those sodium channels to open that are voltage gated. The action potential is rapid. I mean, we saw, a neuron can fire at 200 miles an hour, right? At a microscopic level, boom, 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 opening all these sodium and potassium channels. The interesting part is, too, you can have multiple signals. You can have multiple action potentials traveling down a neuron. Let's say you got a neuron that's 6 inches long, right? Well, you can have hundreds of action potentials that are traveling rapidly down this neuron. Because we're talking about local depolarizations. It's not like you open all the sodium channels and then open all the potassium channels. It's open sodium, open potassium, open sodium, open potassium, open sodium, open potassium, open sodium, open potassium. So this part of the neuron, boom, fires. This part's firing, this part's like hyperpolarized. This part's firing, this part is hyperpolarizing. So you basically have it, which means by the time you get to here, you can fire here again. You can open and depolarize here. Our voltage-gated channels means that you don't have to do anything. Once the ions are flowing in and out, this basically process, channels are opening. So if you are positive outside and negative inside, you're fine. As soon as you reach that threshold, you open your sodium channels, positive rushes in. When you get super positive inside at that peak, potassium channels open to allow now that potassium wants to get out. Because remember, potassium at rest wants to go both ways. It wants to come in because of the concentration gradient, because of the charge gradient. It wants to go out because of the concentration gradient. Once you get all the sodium inside, now it just wants to go out due to concentration and charge. So now you get your potassium out, you return to your resting state, your sodium-potassium pump restores the original distribution of ions, and you're good. Ready to go again. Ready to have another signal come and open those channels. Now, we say this takes time, but we're talking again about, milliseconds, nanoseconds. This is happening very rapidly. However, cells can become overstimulated. When we talk about the synapse, we'll look at specifically how drugs affect the synapse, right? What do they do when you take a drug? Well, certain drugs that people take can actually cause death of neurons. And they do this by overstimulating them. Too much sodium inside is toxic. So if a neuron fires so fast that it can't restore that balance, it can't sodium, potassium pump it out, the neuron can die. Let's look at how some of these things work. How do some drugs work? Will you ever go to the dentist and they inject Novocaine into your gums? They do this in order to, drill and work in your teeth and not have it hurt. Well, they do this by blocking your sodium channels. If your sodium channels don't open, no pain signal. So you cannot send a signal to your brain that says, ow, this hurts, because you're blocking the sodium channels, which makes it impossible for those neurons to depolarize. Now, once an action potential starts, it goes. So once you have this all or none law, once you reach threshold, your neuron's going to depolarize and repolarize all the way down. That charge is going, right? The horse is out of the barn. Once you open it too, you can back propagate. So if a neuron's firing faster, you can actually create a stronger connection between neurons because you're diffusing in two directions. You open these sodium channels, your signal's coming this way, axon hillock, open sodium channels, sodium's rushing in and diffusing, but it's diffusing both ways. So sodium can actually cause these things to become more sensitive. And once the neuron fires, it's going to fire, and it always fires at the same intensity. You can change the intensity by changing the balance of ions around it. That was some early work in studying these things. It was like, what happens if we change the amount of sodium, the amount of potassium? Does it affect the firing of these things? Well, there's all different types of neurons, all different types of ways that they fire. These numbers here on the side, the -70, +40, that comes from those like 50-year-old studies of, even more than that, 70-year-old studies of squid. So we're not worried about this. What we're worried about is these terms. Resting state, you are polarized. Threshold, when you hit threshold, you open everything. When you reach the peak, you open your sodium channels. When you reach the peak, you open your potassium channels, and then you go down back to a refractory period in the resting state. You've got two types of refractory periods, absolute and relative. In the absolute refractory period, your neuron cannot depolarize no matter what. Your neuron is at rest. In the relative refractory period, your neuron can depolarize, which takes an extra strong stimulus to depolarize it. That's the idea of overstimulating. Neurons want to maintain balance. They don't want to overstimulate. There's some interesting things that happen. Now, there's something called tryptotoxin, which comes from the pufferfish. It's also found, it's the fugu delicacy that people eat, this pufferfish. And if it's prepared wrong, you die. Which, I'm telling you, that's a gamble with food. And I'm not sure that's for everybody. The blue-ringed octopus also has this toxin, right? And we know that can be deadly too. And what they do, often if they get people on a ventilator immediately so that their body can be kept alive while it clears this toxin. Now it acts on sodium channels. And so you're talking about, think about, you're injecting the Novocaine into your gums to block your sodium channels so that they can work on it. But now you're talking about your whole body. No sodium channels, no neuron impulse. Your heart needs that to beat. Poisons work in different ways, but some of them can work directly on the ions and the ion channels themselves. If we think about the propagation in a motor neuron, the action potential begins at the axon hillock. It's the swelling where the axon exits the soma, and this propagation term is how it moves down. Right? The idea is you're going to open up, open, close, open, close, open, close as you move down the axon. And this is called, again, saltatory conduction, which is that jumping. Saltatory conduction means you're not opening up channels all the way down, I have myelin here, so I can open sodium channels here. It's going to diffuse to here, open sodium channels here. It's going to diffuse to here rather than having to open more. There is a disease called multiple sclerosis where you have an autoimmune disorder that attacks the myelin-producing neurons, killing them. And what happens is you cannot produce the same kind of action potential. So your body, you can lose movement disorders, vision disorders, because it disrupts the standard neuron firing. The myelin is important to conserve energy and also to synchronize. You need to have a certain speed of things. If information starts getting in different orders, your brain can't make sense of it. All right. What flows in what direction to cause the neuron to depolarize? Potassium in, potassium out, sodium in or sodium out. And the answer is sodium flows in to the neuron to cause depolarization. The what period is when a neuron can depolarize, but it would take an unusually strong stimulus. And the answer is the relative refractory period. Now, here we're going to look at our upcoming things. We've got the synapse. So now we've got an idea of how the neuron works. So make sure you can kind of visualize this process, look at videos showing how a neuron fires, and walk it through to yourself. One of the things you're going to have to be able to do is, in your own words, write down how the neuron fires. We have to address sodium, potassium, refractory period. We have to address threshold, resting state, voltage-gated channels, diffusion, myelin. Okay? A lot of things to consider. So remember that that's the key. And when we talk about the neurons, we're talking about the synapse, the space between the neurons. We get into neurotransmitters and drugs. Thank you very much, and I look forward to next time.